it's a great pleasure for me to introduce Ariane, actually from my lab. She is trying to elaborate approach, a testing approach. I would, yeah, kind of flapsic say the one size fits all test. So if you look genome wide on the tumor cell-free DNA and use bioinformatic pipelines for analyzing this, can you elaborate on testing assay that fits for more tumor entities and gets us a bit off these targeted approaches, which can be tedious if you have to implement them for each patient individually. So Ariane, well, we are curious about your project.
Okay, thank you for the very nice introduction, and I'm happy to present this project to you now. Yes, I think in my talk as Elke introduced already very nicely, I will talk about the potential of liquid biopsy analysis beyond the targeted analysis of single variants. And yeah, I think all of you have seen those kind of clinical applications for circulating tumor DNA analysis today, or have right about an idea. However, I'd just like to say there are different points to make, or steps during clinical progression to address, like, for example, residual disease and recurrence detection, what we've heard extensively today in the first session, and also the treatment monitoring, actually monitoring response or resistance to treatment. And however, one topic that's always coming up again is also cancer screening. What is possible with circulating tumor DNA? To basically, in the beginning, I'd like to say again, how do we actually detect circulating tumor DNA, or what is possible or required to analyze it? And in general, all circulating tumor DNA analysis aren't specifically analyzing ctDNA, but analyzing total cell-free or circulating free DNA, and based on cancer-specific alterations, circulating tumor DNA is detected in the end. And the most well-established approaches to detect ctDNA are targeted ctDNA analysis to detect hotspot variants. Those can be, of course, ddPCR or digital PCR assays, and also NGS assays. But unfortunately, not all tumors cover such hotspot variants, and therefore those assays are limited to those patients that actually have hotspot variants. And to overcome this, untargeted ctDNA analysis are very interesting, because those can be used to track chromosomal rearrangements, copy number alterations, DNA methylation, chromatin signatures, and global and regional fragmentation profiles. And this tumor-specific alterations are present actually in almost all tumors, and therefore are very promising for a one-size-fits-all approach. And within our lab, we established a approach called LiveCNA, or in long, I think, liquid biopsy fragmentation, epigenetic signature, and copy number alteration analysis. It's a whole genome sequencing-based approach. And with this, we actually try from whole genome sequencing data to infer specific ctDNA features or markers specific for circulating tumor DNA. And with this, we first look, for example, on data we get on the fragment length of circulating free DNA and circulating tumor DNA, because it is actually quite nice in closed chromatin, as it's represented here. We expect longer CFDNA fragments, and in open chromatin, we expect shorter CFDNA fragments. And similar, we observe higher sequencing depth in closed chromatin than in open chromatin. And since the chromatin structure is altered in tumors, this can be actually used to detect ctDNA. With our LiveCNA pipeline, we analyzed the global fragmentation profile of CFDNA, the regional fragmentation, CSC-specific chromatin signatures, and somatic copy number alterations. And because all these features are different in cancer patients than in healthy individuals. And to even assess whether we could improve the sensitivity and specificity for ctDNA detection, we use data obtained for global and regional fragmentation to train a machine learning algorithm. But right now, I'd like to show you some of our data we obtained. So first, looking at the global CFDNA fragmentation as an indicator for circulating tumor DNA. As mentioned before, the natural fragmentation of CFDNA is dependent on the chromatin structure of a patient. And when talking about global fragmentation, I'd like to emphasize this profile here. Usually in healthy controls, as this dark blue line shows, we would expect a peak of CFDNA fragment length at 167 base pairs. However, in cancer patients, so for example, in our study, we focused on colorectal cancer, actually, we would expect to have a shift to shorter CFDNA fragments. And with this shift to shorter CFDNA fragments, this would be one tool to detect whether a patient has ctDNA present at a certain time point or not. And when looking at the data of our initial study, we analyzed 55 healthy controls and 134 samples of colorectal cancer patients with tumor evidence. We observed, as you can see here quite nicely, a significant enrichment of short CFDNA fragments from 90 to 150 base pairs in colorectal cancer patients compared to the healthy controls. Then when looking more deeply into the regional fragmentation, so actually also looking at the chromosomal positions where I observe longer CFDNA fragments or shorter CFDNA fragments compared to healthy controls, we summarized it per chromosome arm here, and in green we have again the healthy controls, and purple the samples collected from patients with tumor evidence. And we have, as marked here now with the arrows, several chromosome arms where we've observed significant differences in CFDNA fragmentation. I'd like to emphasize here that we only analyze colorectal cancer patients, so the differences in chromosome arm-wise fragmentation profiles might be different for other tumor entities, but still it might be applicable since it's all compared to the reference set of healthy controls. Then as a third feature with LiveCNA, we analyze CSC-specific chromatin signatures. And in this case, we don't look at the fragment length distribution obtained from whole genome sequencing data, but actually the sequencing coverage. And we use a set of regions known to be active or open in colorectal cancer or described to be open. And when sequencing healthy controls in those regions, we would expect to have a constant coverage, like here in the dark blue lines. And when sequencing CSC patients, we would actually expect to observe a drop in sequencing coverage, because in those patients, we would expect to have those regions active. And we have also seen this in our data when looking at it. So here in green, we have just an example of healthy controls. And those are just two of those region sets we analyzed. And as you can see here at diagnosis, there were multiple patients where we've seen those significant coverage drops indicating the presence of circulating tumor DNA. And when looking at samples collected from patients in partial remission, actually those drops almost disappeared. And in progressive disease samples, we've seen those again. So specifically for the detection of ctDNA based, or the detection of ctDNA based on active chromatin in samples or patients with clinically evident tumor burden is possible, or another possibility. And then we also didn't only want to focus on those non-genetic alterations, but actually also genetic alterations, because as Ellen mentioned in her talk already this morning, somatic copy number alterations have actually a clinical implication, for example, also for the treatment for patients. And this analysis, of course, is also based on sequencing coverage. And this plot is quite complex, but what I would like to show you first as a structure here, we have each block is sorted by stage of the patients, and each line is one patient and each column is one chromosome. And here in those 3 chromosomes marked, I'd like to show you here that in green we have an overlap in somatic copy number alterations detected in liquid biopsy and tumor. And this is already possible in stage 1 patients. So with this pipeline, we were able to increase the sensitivity. And then for further analysis or for assessing whether machine learning approaches could even help to improve the sensitivity for ctDNA detection, we used the data obtained for global and regional fragmentation to train a machine learning algorithm. And also here we obtained really promising results. So we trained a machine learning classifier based on the global fragment size distribution, one on the regional fragmentation, and a meta learner combining the results obtained for both machine learning classifiers. And we've obtained areas under the curve from 93 to 95%, which is really impressive. Just for you as a comparison, when looking at the area under the curve for breast cancer polygenic risk scores, we have achieve a area under the curve of 64%. But with LiveCNA, we actually don't want to analyze only those specific ctDNA features, but actually we want to assess whether performing such a multimodal analysis and combining those features could increase the sensitivity. And here again, I have for each line one patient sorted by the stage of the patient or disease stage of the patient, and each column represents one of the features analyzed with LiveCNA. Here on the left side, we have the tools actually used just for bioinformatics analysis. And here on the right side, and with the purple background, we have the results of the machine learning classifiers. And what is easy to see is that in case of regional fragmentation, so this column here, we achieved the highest sensitivity for ctDNA detection. However, there are some patients where we didn't detect ctDNA based on regional fragmentation analysis, but when combining all features analyzed, we were able to obtain positive ctDNA analysis results for these patients still. And overall, at diagnosis, we were able to detect ctDNA with the basic bioinformatics analysis in 81% of patients, and the bioinformatics analysis in combination with machine learning classifiers in 93% of patients. In localized disease and in metastatic disease, we didn't observe any difference anymore between bioinformatics or LiveCNA alone or LiveCNA in combination with machine learning classifiers. Here, in both cases, we achieved a sensitivity of 94% at diagnosis. And in addition to just ctDNA detection, we also wanted to see whether such an approach would be applicable actually for disease monitoring. And therefore, we analyzed all features. So as seen here, each plot is one of the features analyzed. And this green arrow marks the point of this one patient where for the first time with none of the features, ctDNA could be detected anymore after initiation of chemotherapy. And this was already several months before actually partial remission was clinically confirmed in this patient. And then again, several months before the patient went into progression or had progressive disease diagnosed, for the first time in one of many consecutive samples collected afterwards, ctDNA was detected again, and progressive disease could be actually also clinically confirmed. So overall, we were able to detect response to treatment with LiveCNA in 77% of patients. And when measuring CEA as a reference marker, this was only possible in 25% of patients. And tumor progression was detected with LiveCNA in all patients we included, and CEA also identified this only in 80% or 4 or 5 of those patients. So when summarizing again, what is LiveCNA actually useful for, or what can be performed? So when talking about treatment monitoring, as we've seen just in the slide I've shown you before, By tracking changes in CFDNA features in follow-up samples, it is actually possible to predict response or resistance to treatment. And when looking at in terms of screening or residual disease detection, actually distinct cutoffs or significance tests for ctDNA detection based on the reference set of healthy controls help to specifically identify ctDNA. So far, we established live CNA on a cohort of colorectal cancer patients, but we are currently in process to extend it to other tumor entities, as it is easily extendable because all analyses are just based on those healthy controls. And we are trying to detect differences specific for the distinct tumor entity. Then with live CNA, so the analysis of global fragmentation, regional fragmentation, CSC-specific chromatin signatures and somatic copy number alterations, we were able to develop a highly sensitive and specific approach for ctDNA detection. And with implementation of the machine learning classifiers, we were able to show high sensitivity, but still the training and testing cohort was very small. Therefore, actually larger training and testing sets and external validation might be required for this part. With this, I would like to thank everyone who was involved in this project and supported also me within through the way. And I'd like to thank you for your attention, and I'm happy to answer your questions. Thank you.
Well, Ariane, many thanks for your nice talk, very structured. I don't have questions because I know this project from the back to the forth by heart. And there are no questions in the chat. Yeah, well, it's a first try to figure out if this approach is possible at all. We did it for colorectal cancer first, and we now want to see in pilot projects if it's possible to do it also for other tumor types. And it's at the very beginning. Training cohorts need to be large for all these different cohorts, and it's definitely work in progress. There is one question from Ellen.
Yeah, it's a question or more of a comment. So Ariane, I know everyone is now doing fragmentomics and things like that. How easy can such an approach be transferred to another lab? What do you think? I mean, in terms of pre-analytical conditions, if I use a different type of extraction kit and I use a different type of library preparation, do you think I will end up with the same data in my lab if I do the same?
Actually, I'm not quite sure, but regarding the pre-analytical conditions, we are currently in progress of conducting a preliminary study on this, specifically for the DNA, so beginning with the DNA isolation and the DNA isolation kits. We have a collaboration partner who isolated CFDNA in their lab and sent us plasma samples just to compare whether we get the same results. But I think we will get the results next week. So unfortunately, I was not able to include this yet. I think it's very difficult, and it is really important to test this and not just transfer it. And also, I think the beginning would be also from our side to first test what would be the impact if we use a different library preparation kit. Can we still get the same results for healthy controls, for example, so that we don't detect ctDNA? And can we still, when repeating probably samples with both library prep kits, do we see the same? I cannot answer it yet. I believe it will be quite sensitive, but my hope would be, because fragmentomics, actually the nice thing about CFDNA is that it is fragmented from the beginning, and we aren't sharing it anyhow. So usually I hope that the CFDNA fragment length isn't impaired from the library prep kit, but that's just a hope. It can be quite sensitive to changes.
I think so too, because you always have a somehow introduce a bias, whether you enrich for the smaller ones or the larger one. But we know this issue. Thanks a lot.
Yeah, we've gone through this issue once we were starting to do expression analysis. Yeah, it was a mess in the beginning, but at the end it did work.